Hello everyone, welcome back to my YouTube channel. In this video we will be understanding the hypersensitivity to you can see the key features of antibody mediated type 2 hypersensitivity reaction. So what is a hypersensitivity type 2, also known as the cytotoxic hypersensitivity, is one of the four types of hypersensitivity reaction. It involves immune responses, specially mediated by antibodies, the particular that is the IgG and IgM antibodies. So this antibody directed against antigen present on the surface of the cell or tissue. Okay. So now we understand the antibody mediated type 2 hypersensitivity reaction. So we can see the key features of antibody mediated type 2 hypersensitivity reaction, which are include first, that is antigen recognition. So in this first, immune system recognize specific antigen present on the surface of host cells or tissue as foreign or abnormal. Here you can see in the picture. So how do T cell recognize the antigen? Okay, so first that is a T cells in microbes. Okay, so first this T cell recognize the antigen, right? So the immune system, for example, the T cell recognize the antigen. Okay, so once the antigen recognized by the immune system, now second step, that is the antibody production. Right? So B cell, as you know, the B cell produce the antibody, typically IgG and IgM in hypersensitivity too. Okay, so they are specific to the antigen on the host cells. So there are two types of B cells. One is the namely plasma B cells, and second it is the memory B cells. Okay, so in this picture you can see here it is the B cell receptor. Okay, this is the B cell antigen, the bacterium, right here it is. Now this activated B cell undergoing clonal selection. Okay, so this is the B cell and T cell receptor. Okay, helper T cells which stimulate the production of the memory B cells and plasma cells. Okay, so the main function of the plasma cells is to produce the antibodies, and the memory cells is capable of storing the information of antibodies, producing in case of further attack or during the secondary immune response. Right, now next it is third step, that is the antibody binding. So once the antibody production, that binds with a particular host. Okay, so it is the antibody binds to the antigen on the surface of the host cell and forming immune complexes. Here in picture, that is the antibodies, and this is a B antigen which are present in the blood cells. Okay, blood cell surface, and they form the antibody antigen complex. Okay, so here it is the particular antigen that binds with a particular antibody. Now fourth, death of the antibody bound cell by three distinct mechanisms. Okay, you can say the cell destruction. Here it is the okay, so first antibody binding can trigger the various effector mechanism. First, complement activation. So in this what will happen, so the first, certain immunoglobulin subclasses, okay, so can activate the complement system and creating pores in the membrane of foreign cells. So it is the that is antigen antibody complex. Here example, that is IgG. Okay, they activate the complement system. C1 complex, they split into C2b and C4b fragments. Okay, this is the classical pathways. Okay, now C3 convertase, this is the enzyme. Okay, work is enzyme. Alternative pathways, so the C3 hydrolysis and break down the C3b and C3a fragments. Okay, C3 complement hydrolysis, that is all other one type of protein. Okay, so C3b cleaves the C5, here it is a pink color, into C5a and C5b. So the C3b, which is cleave with this C5 protein. Okay, and cleave into this C5a and C5b. So then they cells swells and burst. Okay, so C5b, C6, 6, 7, here it is C5b. Okay, C6, C7, C8 and C9 together form the cylindrical membrane. Here it is the and attack complexes. Okay, so how the antigen antibody complex, they activate the complement activation. Okay. Now second one, that is the opsonization. So in this opsonization, antibody, okay, the bound to foreign cells also can serve as opsonization, right? So what will happen, antibody coated cells, right? Here it is this is the For example, this is the antibody bound cells. Okay, FC receptor or C3b receptor, right? So the macrophages, it is a macrophages as a work as a phagocytes. So the antibody coated cells may be recognized. Okay, here it is, for example, this is the antibody recognized cells. Okay, that is the antibody bound cells, and which is recognized by the macrophages. Okay, you can also say the phagocytosis. So phagocytosis, there are example of cells, macrophages and neutrophils. Okay, so recognize this through the FC receptor. For this is the FC receptor. Okay, so the antigen antibody bounding cells, right? So this antibody bounds with the FC receptor, macrophages, okay, bound the FC receptor and leads to the cell destruction by oxygenation process. Okay, so this mechanism is resemble as in hypersensitive one, mast cell degranulation. Okay, so this for this understanding you can watch my hypersensitive one IgE mediated cell lysis, okay, video. So link given in the description box, so please watch. Now next step is cell destruction or dysfunction. So in this, antibody can mediate the destruction by antibody dependent cell mediated cytotoxicity. So in which cytotoxic cells, for example, bearing the FC receptor, okay, binds to FC region of antibody on target cell and promote the killing of the cells. So it is the target cell. Okay, so eosinophil also binds with this receptor. Perforin, okay, granzyme B, macrophages, monocytes. Okay, so this is antibody dependent cell mediated cytotoxicity. Okay, so neutrophils binds with this antibody and leads to the cell destruction, dysfunction. Okay, as well as the NK cell also. Okay, that is the target cell. Okay, cytotoxic cell. Okay, so the target cell that present antigen which binds to the antibody and form the anti-antibody complex, and another cells that binds to the with antibody with the FC receptor and perform the perform the cell degranulation, okay, and also leads to cell destruction. Now we understand the autoimmune diseases. So this can occur, for example, autoimmune disease where antibodies target receptor or structure protein on host cell leads to the impaired function. So first, that is the clinical manifestation. So the clinical manifestation of type 2 hypersensitivity reaction depend on the target cell or tissue involved and can range from mild to severe. Critical. Okay, so including first, that is a hemolytic transfusion reaction, so in which antibody detected against blood group antigen on transfused red blood cells. Then hemolytic disease of the newborn. Okay, so in the next lecture we will be understanding hemolytic disease of newborn and hemolytic transfusion of reaction, okay, in next video. So Good-Fasher syndrome, then antibodies targeting the glomerular basement membrane in the kidneys and lung alveoli. And myasthenia gravis. So myasthenia gravis, that antibody against acetylcholine receptor at the neuromuscular junction. Okay, so the next is the Graves disease. Okay, so antibody against the thyroid stimulating hormones receptor and leading to the hypothyroidism. Okay, so all are the autoimmune diseases. One is left autoimmune hemolytic anemia. Okay, so this is particularly antibodies targeting red blood cell antigen. So this is all about today's videos. Okay, I hope this will help you. Thank you for watching. Okay, like, comment, subscribe and hit the bell icon. Thank you for watching.